And now I'd like to introduce you to our speakers today, Professors Rita Popat and Professor Kristin Sainani. So Professor Rita Popat is a clinical associate professor in the Department of Health Research and Policy and the co-director of the master's program in clinical research and epidemiology at Stanford University. She has a PhD in epidemiology and also holds master's degrees in biostatistics and physical therapy. Rita has taught several methodology and data analysis courses at Stanford for more than a decade and has received several Excellence in Teaching and Mentoring Awards from the graduate program in epidemiology and at the Stanford Medical School. Dr. Popat specializes in teaching and mentoring medical and allied health professionals. She has a strong interest in developing curriculum and implementing new instruction models, and recently developed a blended course in epidemiology and biostatistics for Stanford medical students. She is also interested in neurodegenerative diseases and studies the role of genetic and environmental factors in Parkinson's disease and lateral sclerosis. So Professor Kristin Sainani is an associate professor here at Stanford University. She also co-directs Stanford's PhD program in epidemiology and clinical research. Kristin teaches statistics and writing and performs research in sports medicine. She also writes about statistics and science for a range of different audiences. Professor Sainani has received several teaching awards, including Stanford's 2018 Biosciences Award for Excellence in Graduate Teaching. She has authored the health column Body News for Allure magazine for a decade, and she is also the statistical direct editor for the journal Physical Medicine and Rehabilitation. And she authors a statistics column, Statistically Speaking, for this journal. She teaches two popular massive open online courses, including Statistics in Medicine on Open edX and Writing in the Sciences on Open edX and Coursera. And now I'd like to turn the floor over to Kristin and Rita.
Thank you. Well, welcome everyone. Good to have you all. So what I'm hoping to do in the next few minutes today is to give you an idea of the kind of content and material you're likely to see in the certificate program in clinical research and epidemiology. So just briefly, when we talk about clinical research and epi, what we're often interested in is in understanding or evaluating a relationship between an exposure and an outcome. And the exposure could be anything. It could be a genetic factor, a non-genetic factor, a drug, or any sort of lifestyle factor. So here are three examples that just hit the news last week. So I kind of researched this in preparation for today's webinar. So here's the first one. Does alcohol consumption during breastfeeding, is that associated with poor cognitive outcomes in children? Another example that we saw recently last week was actually researchers here at Stanford, where they examined whether acetyl L-carnitine, I'm just going to call it LAC, whether these levels, and this is a chemical, by the way, which we all produce naturally in the body, and it's important for brain function. And they wanted to see if this was associated with increased risk of major depressive disorder, which I will just refer to as depression. And here's a third example. This is sort of a stem cell therapy example where do induced pluripotent stem cells, can they help sort of cure, so to speak, or reduce the symptoms, alleviate the symptoms in Parkinson's disease? So as you can see here, we've got three examples of three very different kinds of exposures. We've got alcohol consumption, which is, of course, a lifestyle factor. It's an exogenous sort of risk factor. We've got a naturally, endogenous chemical like the LAC. And then we've got sort of stem cell therapy. Let's elaborate a little bit. Let's look at this example with LAC levels in MDD. So as I said, these are researchers from Stanford. The study was published in the Proceedings of National Academy of Sciences. So this got picked up, got a lot of news attention. You can see there's one article talking about whether low LAC levels is a new biomarker. So with this new interest in precision medicine and the role of biomarkers in sort of predicting who's at risk for certain diseases, this is sort of an interesting angle to look at this particular chemical with. And then we've got a blood test that could be used for treatment. So you've got a prediction sort of potential as well as a treatment potential. Well, very briefly, what the researchers did is they looked at lac levels in the plasma among a group of patients who had MDD and a group that didn't. So the blue dots are the ones who don't have major depressive disorders, and the red dots stand for people who actually do have major depressive disorder. And you can see that lac levels are lower in those with MDD compared to those who don't have it. And we won't get into the statistics, but you can see it's a highly statistically significant p-value of 0.0002. So you might say, oh, wow, should you be compelled? Should we all go out and start taking the supplement, which, by the way, is available in sort of health food stores? So would you be recommending this to your relatives or friends who may have MDD? I hope you all will be skeptical, because before we see and decide whether we should start taking this supplement, we want to ask the question, and this is what we hope to train everybody who's in our program, to first ask the question, well, how was this association between lac levels and MDD evaluated? So we logically think of this as 3 steps. The first step is a research methodology. Then we go to step 2, which is how was the analysis done? And finally, how do we make now our interpretation based on the results and how the study was conducted? So let me elaborate a little bit on step 1, since my colleague, Dr. Sainani, will elaborate on step 2, which is the statistical analysis. So in step 1, when we talk about research methodology, simply put, we're asking 3 basic things. How was the study done? So what was the study design? Who was in the study? So what was the study population? And finally, what measurements were made? So I want you to think about, okay, well, if you were interested in this relationship between lac levels and MDD, what do you think might be an appropriate way to actually design this study? Now, I'm going to leave out the clinical trial because, we're not going to start randomizing people. We first want to see evidence from observational studies. So, okay, let me help you out. And I'm sure you all, this is, you can probably come up with this design. Let's say you're the investigator. You're going to get a group of people who are don't have MDD now. And we won't get into inclusion-exclusion criteria, but you will choose your cohort of people. You will then measure their lac levels at baseline, and then you will follow them forward in time. And then you'll evaluate who develops MDD versus not. Sorry, guys, I know the guys lying flat are not meant to be dead. It's just to, illustrate the 2 different groups, a group that developed MDD and a group that didn't. By the way, a fancy word for this design is a prospective cohort study. And we like these studies, and you can see why, because causality is in the right direction. What I mean by that is that the exposure, that is lac levels, comes first, followed by the outcome MDD. So if we do find an association, at least the design helps us explore whether this is causal in nature. As I mentioned, that's a fancy term for this design. Now here's another way to do it, and maybe some of you came up with this design. So you're still the investigator, but now you start with a group of people who have MDD and a group of people who don't have MDD. And then you go back and then you try to determine their lac levels, either taken at that cross-section in time or maybe from samples that were stored previously. Now let me ask you this. Among these 2 designs, oh, by the way, this is the case control study. So among these 2 designs, which one do you think is a stronger one to sort of bear more credibility that there is a causal link? Well, I think we would all say the prospective cohort study. What if I told you what these researchers did was actually a case control study? So you see, before you get all starry-eyed and blown away by the statistics and the p-value, you really have to evaluate how the study was done. Now, I'm not trying to say that the researchers say that there is a causal link, so I want to be mindful that they, were correct in the way they interpreted the data. But for most of us, when we look at the literature, we just focus on the statistical piece and the p-values, and less so on how what the methods were that generated these results. And in my courses, I focus a lot on this methodology piece because we can see that certain study designs lend themselves better to understanding causal associations. We also want to ask other questions. So this is about measurements. How was major depressive disorder ascertained? Were reliable and valid methods used? Was it used in a standardized manner in all in the entire study sample? How were the lack level measures? Were the assays reliable? How about validity? Does a lack level taken at one cross-section of time, is that truly reflective of one's, exposure to these lack levels over time? So you see, it raises a ton of questions which we need to keep in mind in order to correctly interpret the study findings. And then because it's an observational study, we worry about other confounders as well. So what we have to really be able to separate is causation versus association. We can see non-causal explanations that can generate spurious associations between an exposure and outcome. So in this course, we also spend time talking about sources of study biases that can produce these spurious associations when actually there's nothing going on between the exposure and the outcome. So we first have to actually rule out all non-causal explanations and then ask the question, okay, well, is there evidence to support a causal association between the exposure and outcome? And two quick points. Just because you observe a statistically or a clinically significant effect doesn't mean there is a real effect. And the converse is also true. Just because you fail to observe a statistically or a clinically significant effect, it doesn't mean that a real effect is absent. The study methodology could be so flawed that you actually fail to pick up a signal. A flawed study methodology can also do, what we see above, where it can produce a spurious effect. So we kind of have to be detectives, clinical research detectives. And what, as detectives, we do is we try to figure out how much was a study finding influenced by bias. And what's bias? It's any deviation of truth beyond chance error. So these are systematic errors by the investigators in sampling, collecting, or interpreting data. So this is how we sort of culminate the course, where we've learned about the methodology, and I give students papers to read. They read it, we look at the study findings. If we believe that the study has some truth in it, then we say the study has internal validity. And then we move on and we ask, oh, could this be useful in clinical practice or in designing sort of the next sort of research question? On the other hand, if we determine that there's too much bias in this study and that it really affects the validity, and we are skeptical whether there's truth in the study, we stop right there. But in the previous scenario, if the study does have some truth, then you can ask about generalizability or external validity. And this is where we ask the question, can what we found in our sample be applicable to the study population beyond that, and where we want to apply these findings to? But if we messed up the way we sampled and the way we actually studied and came up with the study sample that generated the results, then while the study may be valid, it may not be generalizable to the population beyond that was included in that particular study. So true or false? I get this question a lot because not everybody is going to be doing prospective data collection and be designing these studies, and you may be working with existing data. So here's the question I get. We don't need to worry about study design and measurements because we're using an existing database or a data source, and information has already been collected. We just need to understand how to do the data analysis. Of course, I'm setting you up because this is false. And I hope we've given you I've been able to give you a small example to show you why it's not, even if it's just secondary data that you're using, you still within that need to ask yourself about measurements, about whether it's the right database to be answering a particular exposure outcome relationship, and whether you can even get to answering the question about causation. So in a nutshell, when we want to examine an exposure outcome relationship, step one is research methodology. The statistics is very important too, because epidemiology and clinical research is a quantitative field. So you have the methods and you need the statistics and the analytic approaches to be able to quantify the relationship between the exposure and outcome. But the methodology informs the statistics. And in this series, in the certificate program too, you will learn about statistical approaches that can actually help you, account for some of the weaknesses in the study methodology, things like confounding and control for those types of variables. And finally, we need both the methodology and the analysis to help us with the interpretation. So clinical research and epidemiology, for whom? Who should be studying this? So if any of you are interested in research or studies that involve human subjects, either active data collection or recorded data, this might be the one for you. So what skills will you learn? These are just some that, I've put here. I'm sure there are others, and Dr. Sainani can chime in. But here are some of the skills that you hope students, not just in our program, but in our certificate program, will learn. How to critically evaluate a published paper. How do you conduct peer review of a research manuscript or a grant? So some of you may be serving in these roles, and it's a huge contribution to the scientific community to know how to critically, do a peer review of a manuscript or a grant, which is all done on a volunteer basis, by the way. How to develop a study protocol for an observational study or a clinical trial. So for those of you who are in the trenches, who are helping PIs design studies, conduct studies, our courses will help you develop these type of study protocols to assist the PI, or perhaps you are the PI who's developing the protocol, to help you develop an analytic approach appropriate for the proposed study design, to conduct data analysis, of course, with the help of the courses that my colleague here, Dr. Sainani, teaches, and how to interpret results. So very briefly, in our master's program, where we have 3 core courses in the methodology series, you're seeing that the numbers here, HRP 225, 226, 251, the description is given below. In the certificate program, we're asking students to just required to take 2 of them. That's 225 and 251. So what do these courses cover? HRP 225, which is Design and Conduct of Clinical and Epidemiologic Research Studies, we cover descriptive epidemiology, sampling methods, reliability and validity of measurements, analytic study designs, their strengths and limitations, so sort of cohort, case control, cross-sectional studies. We talk about measures of association, things that you might be familiar with, like odds ratios, risk ratios, hazard ratios, sample size estimation, how do you power your studies, how many people do you need to study a relationship, sources of bias, confounding, we talk about interactions, we talk about causal inference. We do a systematic critique as a culmination of having learned all of these concepts, and we also touch upon diagnostic tests and screening. The spring quarter class, which is called HRP 251, is focused on clinical trials exclusively. So you must have an intervention that you can randomize. Now we also touch upon early phase trials, so let me just go through some of the topics that we cover in this course. We've got design issues, how to think about outcomes and how to select them, surrogate versus hard clinical endpoints. We discuss phase 1, phase 2 studies. These are your early phase study designs, phase 3 study designs, the large efficacy trials. We talk about recruitment and adherence, sample size and power. Since we're at Stanford and we have people who have developed amazing adaptive designs, so you will hear about adaptive designs in designing your clinical trials, how to handle missing data, data analysis, and interim monitoring, both from a statistical perspective as well as a data monitoring committee perspective. So with that, I hope I've piqued your interest and your appetite for clinical research and EPI. And now to my wonderful colleague, Dr. Sainani.
Thanks, Dr. Popat. That was actually a great lead-in to my presentation because my courses focus primarily on the statistics, but it's so important to keep the statistics in context. How were the data collected? Where did those data come from? You can't interpret the statistics in a vacuum. So I teach a 3 course sequence in biostatistics for the master's program in epidemiology and clinical research at Stanford. You see the course numbers up there. For the certificate program, you would take the first 2 of those courses, which is my introductory and my intermediate biostatistics courses. So I'll just start by talking a little bit about something that Dr. Popat alluded to, which is that you have to be skeptical of the published literature. And we have, as many of you are probably aware, a reproducibility crisis in science. And this has to do with many facets, as Dr. Popat alluded to. Research design, how you design the studies is a key part of that. I'm going to focus, of course, more on the statistics, but that's it's all important. This is one of my favorite studies that came out in 2005 by John Ioannidis, who is actually now a faculty member in the Division of Epidemiology at Stanford. And he published a paper called Why Most Published Research Findings Are False. And he was pointing out that a lot of the findings in the literature actually are false positives, spurious associations that can arise either because you do a study with bias, or because of the study design, or because of the statistics, or all three of those. If you like late night television, as I do, you may have caught, there was a John Oliver piece a couple of years back just dealing with talking about this whole reproducibility crisis. So you see this even in the media and the popular media right now. There's been a couple of high profile cases I'll just point out here. So there is a case with a scientist, a high-profile scientist at Cornell who does food science. His research had been highly cited, talked about in the media, and his research has come under scrutiny for a number of reasons, one of which is sloppy data handling, something we're going to try to, I'll talk about with you in my classes to try to help you to not have sloppy data handling. Another issue is a lot of his research findings seem to have come out of what's called p-hacking. So what that means is if you parse your data in enough ways, if you kind of try all possible associations with all the variables in your data set, you are going to find some spurious associations, some things that achieve statistical significance. P is less than 0.05. But those are almost certainly going to be false positives. And that's one of the things we talk about in my classes is trying to avoid p-hacking. I'll just put up a few more. This was an article that was in 538.com. This was actually something I was involved in. There was a statistical method introduced into the sports science literature that causes high false positives. Some of you may have read in New York Times or other media outlets about some famous research on power poses in psychology that's been largely discredited. Again, a lot of these things have to do with poor study design as well as poorly done statistics. And so what I try to do in my classes is to teach you how to do reproducible research and to do good statistics. I blame some of the issues with poor statistics in the literature on how we teach statistics. And I'm not saying that all statistical teaching is bad. I think there's some great statistical teaching that goes on out there. But I noticed that a lot of statistical teaching seems to fall in one of two camps. The first approach is a perfectly valid method. This is how I was trained, but it's highly theoretical with a lot of math. So a lot of calculus, linear algebra, and proofs, not so much practical training on how to deal with data. And again, I love this kind of stuff. I like to do proofs. One of those equations up there is from one of my papers from this year. But this approach is not appropriate for every audience. So people who are doing applied sciences and psychology, education, medicine, may not need to know all of the math and may not need to work out all of the calculus if their goal is really just to analyze their data properly. And if you try to teach it in this way and it's not a match with the audience, sometimes what ends up happening, I've noticed, is that we swing to the other extreme. So instead of teaching all the math, we suddenly teach none of the math, and you end up with more of a cookbook approach. And that's an approach that I find really problematic. So because when you end up taking out all of the math, it becomes more of a, I just have some data. I want to know how to get it in the statistical analysis program and get something out, click the right buttons and get results. And I think this is a problematic way to teach statistics and also to do statistics. It leads to some of the reproducibility issues that I was just talking about, because if you are just kind of throwing your data into a statistical black box, not really understanding what's happening in that box and what the results mean that come out, unfortunately, this leads to all sorts of problems. And one of the problems is that if you are kind of fuzzy on what exactly is coming out of that black box, it's too easy to cherry pick out of the results that come out. And I'm not saying scientists do this intentionally. I think it's mostly unintentional. But if you're kind of fuzzy on what those results mean, it's very easy to just kind of select the ones that seem to support the story that you want to tell, and then get excited about that story and publish a story that is misleading. I'll just give you one example of this. This is something I was looking at recently. This is from a published paper, and they were looking at glucosamine, which is a supplement that sometimes people take for knee osteoarthritis, which is pain and stiffness that you get in your knee as you grow older. And they randomly assigned, it was a randomized trial, it was a small randomized trial, I think it was about 50 people, and they randomly assigned half to take a glucosamine supplement daily and half to take a placebo pill. Now, if you looked at the published study, there was no graphical representations of the data. They simply put all the data in a table. And I was looking carefully at that table and noticed something. And so I do quite a bit of statistical sleuthing. As Rita said, we have to kind of be a detective, and I really enjoy statistical sleuthing. So I actually just pulled the data out of the table and graphed it myself. There were two primary outcomes for this study. The pain that you have in your knees and your disease severity, sort of an overall disease severity. So I'm going to show you my graphs. This is the data that I just took out of the tables and I simply graphed it. And what you're seeing here is the graph for pain. The y-axis is the percent change since baseline. You want it to go down because you want pain to get better. And the x-axis is time in weeks. This was a 24-week study. The blue line is the treatment group, the red line is the placebo group. And you do not need to know any calculus or have a degree in math or statistics, I think, to interpret this graph correctly. I think we can all look at that and agree that there was maybe some small decrease in both groups. If anything, the placebo group went down more in pain than the treatment group. And actually, those two lines are sort of within the statistical margin of error of each other. So really, there's no difference showing up between those two groups. The authors actually of the paper got that right, as I'll show you in a minute. They got the right conclusion with pain. Where things went wrong is when it came to disease severity, which is some kind of scale people fill out to say how bad their disease is flaring up. Again, we want it to be lower. To take a second here and look at that graph, same general graphic, treatment is in blue, placebo is in red, and take a second and see what conclusions you think you would draw from that graphic. I think all of us kind of looking at that would probably agree that it doesn't look like there's much difference between treatment and placebo. In fact, they ended up after 24 weeks at almost the same level of disease severity. In fact, you can see there's some drop in disease severity, and this indicates that there's some kind of placebo effect going on. So it's a subjective measure. People take a pill, and when you take a pill, sometimes you feel better just from the placebo effect. So that seems to be what we're seeing here. The only real difference that you can see between the two groups is probably just due to chance fluctuation. They have a slightly different trajectory. So for some reason, might be just chance, the treatment group went up a little before it went down and then kind of was level. Whereas the placebo group went down a little and then up and then went down, but they ended up in the same place. So it might surprise you to hear that what I would consider a null result, in the paper, the authors put a positive spin on it. And in fact, this comes right out of the abstract. They say, these results suggest that the consumption of glucosamine, the supplement, for 12 weeks or longer has a positive effect on osteoarthritis score. Osteoarthritis score is that disease severity score I was just showing you. Now, they got it right on pain. In contrast, it does not reduce pain. So let's look at that graphic again. How in the world is that a positive effect? The placebo group and the treatment group basically did the same. There doesn't seem to be any benefit from treatment, and I think it's easy to see in the graphic. So what happened here? I think this is one of those cases where the authors took their data, they stuck it in the computer program, they were happy to get out some results, and in the midst of all those results, there were a few significant p-values. And I'll tell you, there was a significant p-value. The drop from 4 weeks to 12 weeks in the treatment group happened to be a significant decline. But guess what? The drop in the placebo group from 12 weeks to 24 weeks was also a significant decline. They didn't make a story out of that. But there was a true decline here. There was an actual placebo effect. People did feel a little better subjectively. There was also a significant p-value. There's a technique called repeated measures ANOVA, and when you plug those data into that, you do get a significant p-value. That significant p-value, though, just tells you that the trajectory, the lines, the paths those two groups took was a little bit different, as you can see visually here. It does not tell you, however, that the treatment group beat the placebo group. Unfortunately, if you're not quite sure what these statistical methods are doing, you see those significant p-values, you get excited, you make a story that's false. So I'm going to teach you in my courses some principles of effective statistics. One of the most important things, I think, is that you can't do statistics well without understanding the basics of probability and statistical inference. You can't do statistics without any math. Now, that doesn't mean we have to swing to the extreme of putting in a ton of calculus and linear algebra. I believe that most of what you need to know in statistics can be taught with subtraction, multiplication, division. You may also need to be familiar with like decimals and fractions, but that's the level of math that's required in my classes. You also need to use common sense around numbers. I'm going to try to teach you what is statistical inference, what is probability, and a good, strong understanding of those, but you don't need the fancy math necessarily for that. And one of the techniques that I use for teaching is computer simulation. We will do a lot of computer simulation. So I'll just give you an example of that. There's a lot of problems that can be solved mathematically, but can also be solved in a computer. And so one of the classic probability problems that you're going to see in any sort of probability 101 course is called the birthday problem. And it goes something like this. Some of you may have heard of it before. Imagine you have a class of 30 students. What's the probability that in a class of 30 students that at least two people are going to share the same birthday, like they were both born on August 7th? And instinctively, most of us think that that probably is going to be pretty small, right? There's 30 people and there's 365 different birthdays. Surprisingly, though, that probability is quite high. It's in a class of 30, there's actually a 70% probability that two people will share a birthday. You can solve that problem mathematically, and for those of you who are interested, I will solve it mathematically in my course for you. But you can also solve that problem with computer simulation, and I think the computer simulation is really intuitive. And what you do, we have great computing power these days, so you can do this on a laptop in a second. You actually have the computer randomly sample 30 birthdays out of all possible 365 or 366 if you include leap years birthdays. And you have the computer do that an arbitrarily large number of times, so like 100,000 times. The computer can do this in a second. You look then at all of those samples, and you look at the birthdays and see in how many of those samples was there a match. You have the computer count up in all of those different 30 classrooms, out of 100,000 times, how many times did you get a match? And that will give you the exact same answer as if you solve it mathematically. So we'll do a lot of computer simulation to help motivate what's happening under the hood in statistics. Another principle of effective statistics is you need to understand your data. And so we don't just stuff things into the statistical black box and press the button to do a t-test. Before you do that, 90% of your data analysis is actually not doing the t-test or the linear regression or getting p-values. It's actually drawing pictures with your data, understanding how much missing data you have, understanding where your data came from, making sure that you don't have errors in your data, and really just getting a sense of what's going on in your data before you ever run a formal statistical test. I've already talked about drawing lots of pictures with data. You need to use common sense. Always consider the totality of the evidence, don't just cherry pick. And we're also going to talk a lot about keeping an accurate record in code of everything you do to the data and all the analyses you do. So that I mentioned earlier the case of the scientist at Cornell whose work has come under fire. A lot of the problems have to do with not keeping an accurate record, not being able to produce data sets or code that back up an analysis in a published paper. So I teach, again, 2 courses in the certificate program. In the fall, you'll take HRP 259. That's my introductory course. We will spend quite a bit of time on the theory of probability and statistical inference. A lot of that, as I've already mentioned, we will do with computer simulation, and there will be some math, of course. You have a choice. You can work in either R or SAS. We use the free version of SAS University Edition. R is also free. And you don't have to do the course in both of those. You're just going to pick one of those, and I will teach you how to code. There is no prerequisite. It's fine if you have never coded in either one of those before. if you want to learn both, you're welcome to try to learn both. We will also do the practical side of data analysis, how to think about, look at, and describe data, how to actually get your data into R or SAS, clean it, manipulate the data, count up the missing data, and then how to actually analyze it. We will talk about how to choose the right statistical test, how to run a regression, how to interpret the results from those statistical tests properly. We'll talk a lot about common statistical pitfalls that underlie a lot of the reproducibility crisis in the literature. And I really enjoy being a statistical sleuth, so I will teach you how to be a statistical sleuth. My winter quarter course is a mix of theory and practice again. We will continue to analyze data in R or SAS, getting a little bit more complicated in our coding. I will talk about some more advanced regression techniques and things like how to deal with correlated observations and some more advanced techniques like principal components analysis and propensity scores. And again, we will fundamentally make sure that we're not hitting statistical pitfalls, and also we'll learn how to be a statistical sleuth. And that ends my portion, so I'm going to pass it back to
Great. Yeah. So thank you so much, Kristin and Rita, for this really informative presentation. And I hope all of us who are here with us today can really walk away feeling more confident and knowledgeable about both of your work, and as well as kind of some of the topics that are really related to epidemiology and clinical research. So now let's move on to the Q&A portion of this webinar. So the first question that we have, for the both of you really is really more about kind of, the prerequisites. So not in terms of, courses, do I need to take, Stats 101 or, maybe a senior level stats course back in undergrad, but more so about kind of the knowledge, that you guys think would be most appropriate before coming into this program, so that they can get the most out of these courses. So if you can kind of just talk a little bit more about prerequisites and knowledge, that would be great.
Sure, I can jump in. So there are new prerequisites in terms of you do not have to have had a previous stats course. You do not have to have experience coding in R or SAS. We will teach you all that. As I alluded to, you have to be comfortable with basic math, decimals, fractions, nothing too complicated. And it's helpful, I think, to have some context. So if you are somebody who's doing research and you're familiar with the medical literature, that doesn't mean you have to be an expert on it, but you've maybe read a medical paper or are involved in research. That context does provide sort of the basis and the motivation to take our courses.
Yeah, so for the methodology courses, there are no prerequisites. I think having some familiarity with having at least read an occasional scientific paper is helpful. And if you've taken a stats course, of course, that's helpful. In terms of content, we are within a school of medicine. Our examples tend to be very clinically focused. But we have students all across campus who take our classes. So we'll have students from engineering or, some other discipline who may not know enough about medicine. And when we first bring up examples, oh my gosh, do I need to know what Parkinson's disease is? Like, no, you don't. What's the wiki for? Wikipedia. No, you don't need to know. The principles apply broadly across, and you don't need to be an expert in biology and neuroscience or anything. You can come from pretty much, any background and benefit from this material and be able to get value from the courses.
Great. Well, I hope that was really helpful for all of you. So the second question that we have is, based on your knowledge, what is approximately the time commitment that's related to some of these courses? So, because a lot of our audiences are working professionals or are full-time students. So how much time do you think they would have to dedicate to successfully complete the courses and the workload?
Okay, well, there's always going to be a range.
Yes.
But so I think in terms of, and by the way, for the students or the participants who are asking these questions, you're not alone. In fact, in our master's program, substantial number of students are actually fellows and residents or medical students who are juggling several responsibilities. What we've done is, so in terms of, in-class time, for example, 225 and 251, we meet twice a week. So that's 3 hours of in-class sort of participation or watching the video. of course, there's going to be homework assignments. So I would say in addition to the 3 class time hours, maybe another 3, again, this is the range. It could go from 1 to maybe 5 hours, depending on how familiar you are and how in depth you also want to go. So I think that's.
I would agree that it's, you know, 5 to 6 hours might be per class is probably a good target. You should know that we both, Rita and I, designed all of our courses around a physician audience, so working physicians who have to go to clinics several days a week. So we have designed our courses to be very comprehensive, but also efficient, to help to save you time. For my courses, all of the lectures are online. It's about 2 hours of lectures, including the lectures on programming each week, and then of course, there's some reading that you could do alongside. Sometimes you may need to watch a video twice if it's maybe a more complicated probability question. So it might be 3 hours. And then I also have class time where we spend doing solving problems. So again, it might be about 3 hours of lecture and sort of class exercises. And then however much you want to prepare beyond that, I have a weekly homework. There are some online quizzes. So it might be about 3 hours, I would say it would be a good target. But there will be a range depending on your comfort level with math and familiarity with these topics.
Great. Well, so the question that we have moving forward, and I think this was actually a really popular question from our participants, is a lot of them are actually thinking about maybe using this as a stepping stone into a PhD program. So can you kind of further elaborate on, how some of this content in the course could potentially help them pursue a PhD in the field, and would you recommend this course to students, who are taking it for that purpose?
Yeah, I would think that, I mean, students in our PhD program take some of our courses, these same courses. And let's say that you want to do a PhD, and sometimes people pursue a master's first, but let's say you're don't want to necessarily do a full master's first, you could get a lot of the beginning content through these 4 courses. That would be a good foundation to know if you really want to continue and do a full PhD in this area, and also to have a good foundation to do a PhD. Absolutely.
Yeah, I think it depends, of course, on your background. So if you have, I'm assuming that you want to pursue a PhD in this field, or something that would be related to, that will require these skills, in which case, as Dr. Sainani said, I think getting, does this subject material really is of interest to me? Do I get excited by it? Would be a wonderful opportunity. And then, of course, to also test the waters and see how comfortable you are with the material, and whether you can really challenge yourself, within the domains of statistics and methodology as you apply for a PhD program.
Great. So on that note, we also have a few students and participants here who, are asking about kind of academic help, throughout this process. Since most of these courses are going to be offered online, so will they receive kind of, the same level of academic support from the professors, from the teaching team, and how will that process be kind of carried out?
Yeah, we will treat you as any other student in our courses. So you are considered a student in the courses. We see you on our same enrollment list, and so the additional thing we're going to do for the online courses, because of the issue of being remote, and some of the in-class activities that we do are very hands-on, I think, both for my courses and Dr. Popat's courses. So we will have a TA who is the liaison to the remote students to help make sure that those activities that you're trying on your own, where normally we walk around the classroom and sort of look at your code and, oh, you're missing a semicolon there, that there's a similar experience for the remote students by having a TA who's dedicated to that.
Yeah, definitely. I think we are committed to supporting the students who are doing the certificate program in the same way that we're committed to our students who are on campus. So we will make sure that there are TAs who can provide the help and assistance.
And will kind of office hours also be available for students who are remote?
That's a great question. Yeah, I have a normal Monday office hour, and we may have to figure out how that exactly works, because we won't be in real time with the students, so we will have another office hour option for that, yeah. Also the office hours are taped, so if you wanted, sometimes students just come to get help on the homework, and then often if you have the question, somebody else who actually comes to the office hour will answer the question. So some of my on-campus students actually don't come to the office hour but watch the video, and then they can get the questions answered.
So I don't tape my office hours, but but we'll make sure that a TA is able to respond to your questions, and we'll be available as well.
Great. And then now it's time for kind of a lot of people's favorite question, really about the outcomes, right? Like, what am I going to get out of this program, and what type of career paths and, employment options would be available to me? So just based on your experience of teaching this course here on campus in the classroom, so what type of career routes and paths do students usually take after completing these classes?
So I can take a couple of examples. So I'm talking to now necessarily not the physicians. I don't know if actually we didn't get a chance to see the poll results from the first poll, so I don't know what backgrounds our participants come from. But I assume this question is from those who are not clinicians or clinicians in the making. And I think there are actually two or three that come to mind, three groups. One being, so if you're involved, if you're a research coordinator and you want to do actually more, not just recruit patients and help with the day-to-day functions of a clinic or a lab, then having these skills, you can be more involved in the and assist the PI in actually helping them develop protocols, as well as setting up databases and even helping with statistical analysis, to look at sort of preliminary data and so on. So that's one area. Another area is if you come from the writing world, medical science journals, this would of course be immensely helpful. The third group is if you are, say, working at a biotech company. So Gilead, Genentech, these pharmaceutical companies often have epidemiologists who are on their teams. And often what the epidemiologists have to do is to actually see what's out there, what's been published, what's the disease burden, and they have to actually do sort of a systematic review of the existing evidence, and these skills will help you sort of add to the kinds of things that you can do in these roles.
Yeah, I would just add that I think the skills that we teach are largely foundational skills. I mean, they're in a particular area about medicine, but data science is a huge field right now. Being able to analyze data in R or SAS and understanding the implications of where the data come from, that's a transferable skill across a lot of fields right now.
Right, and if I may add, actually Dr. Sainani just reminded me, so let's say you're someone who just comes from a biostats background, you did a master's in biostats.
Good point, yeah.
I think because often the statistics just sits in a silo, learning about the methodology and how to actually approach the data analysis. What we constantly tell our students is that the statistical program is just a tool, and we don't want people just using it as a black box. So how to think about analyzing and thinking about the approach that's informed by what the methodology was and what the measurements are the skills that we do teach. And actually, Dr. Sainani and I have been teaching this for over a decade, and so I pretty much know what she covers, and she knows what I cover, and it's a very nice complementary sort of teaching sort of partnership.
Yes.
Great. Yeah, and this question is specifically, for Dr. Sainani. So we have a participant that asked about, how rigorous really is the statistics portion of this program? So are we going to be dealing some of the more advanced topics, including kind of imputation and hidden Markov models and such?
So I would say it's very rigorous in the sense of that I want to start in the foundations, and I want you to do data analysis properly. So you're going to document your code, you're going to understand the models. My HRP 259 course largely focuses on that, so we don't necessarily get beyond regression analysis. We focus on doing statistics right, and I actually think that once you have that, the particular statistical test that you're going to run, that's easy to look up online and figure out how to do it in R or SAS. So I think the foundational stuff is much more important. In my second course, we cover some more advanced topics. We don't cover Monte Carlo simulation I cover a little bit about imputation. We talk about missing data imputation. Dr. Popat also talks about that in her courses. We don't get into it in a very, very deep way, but I would say the courses are quite rigorous in that you are expected to not just be able to do those and implement those, but really understand the foundations that build up to those.
Yeah, I think we do have courses in place. So,
Right.
I teach one in the spring, but it's not part of the certificate program. So I think you need to, as Dr. Sainani says, once you have a good foundation, pretty much, and these statistical techniques you have to be aware are going to keep developing. In fact, we have to constantly educate ourselves about what's the new method of doing, X, Y, or Z.
Right.
And once we know the basics and you look up the papers and you read them, and then usually the code is available and
Right. I teach a lot through computer simulation, and I'll tell you, if a new statistical method that I've got to learn, I find, oh, I got to run that on my data. First thing I do is pop it into the computer and run a simulation. So I have a sense of, I have some data which I understand the generation of the data. I put it in this statistical method, then I can understand the pluses and minuses of that method. So the particular methods to me in a way are less important than knowing
Right.
how to assess them and how to understand what's going in and out of those methods.
and also need to know, sorry, this is a pet peeve of mine, because there'll be a new thing like instrumental variables or even propensity score matching. And yeah, these techniques are all sophisticated and fancy, but sometimes it's overkill on the data, really.
Exactly, yeah. And sometimes it's something
Simplicity is
recycled
good.
that actually you can do in a simpler way, and they're just trying to make it fancy. So.
Great. So now I know we're getting a little bit close to 12 o'clock, so we have time for two last questions. So the second to last question that I have here is really about, since this course kind of focused more on kind of clinical, medicine, right? Like just in a more traditional sense, will any of the courses cover kind of alternative medicine and clinical research or integrative medicine?
Via examples. So again, our course is not specific that it has to be clinical or public health. I think the principles broadly apply. If you're interested in community research, if you're interested in alternative medicine, integrative medicine, great. I love it. But you can use the tools to evaluate these, alternative therapies in a rigorous way, so that actually, I think we need to see more rigorous research in that domain. So via examples, you certainly see them.
Right. We certainly have a broad range of students on campus. I think, as Dr. Popat said, I have some from engineering, from psychology, from education. And so for them, the specific examples may not be exactly in their domain, but that's okay. They're applicable across.
Great. So the last question that we have today, and I'm sorry that we still have 50 or so questions, from all of our participants I would love to address, but we only have time for one last one here. So the last question that came from participants is that what type of resources would you recommend them kind of exploring, between now and perhaps September, so that they can kind of prepare for the content for the course? If there are any resources that you would recommend, like books or websites or courses, kind of more intro level courses that they can, seek out outside of Stanford?
Perhaps your MOOC?
Yeah, I mean, I
Statistics
have a
in
MOOC
Medicine.
that's a good introduction, and you could check some of that out as an introduction. You don't feel like you need to do a bunch of homework before you come to us. We start from step one. If you want to download R or SAS University Edition and play with both of those and figure out which one you prefer, or maybe do a little research about different areas of research. People will use SAS and some people will use R. So kind of figure out what people around you are using so you can make that choice. That might be one of the things to do for homework.
Actually, that's a great suggestion. And I am more comfortable in SAS, so that's what I tend to use in my courses. Of course, we won't be using it that much for methods, but SAS University Edition has wonderful tutorials and video, recordings of how to download it, how to use it.
Right.
So it's a good idea to play around.
And again, we'll provide, we have a good colleague right now who is currently in the moment taping some introduction videos on how to download R and SAS. So you don't feel like you have to do that, but if you want to be prepared and do a little bit of that ahead time, it'll just make your first week easy.
Great, perfect. So thank you so much again, Rita, Kristin, for answering all the questions from our participants and for a really great and informative Q&A session. So we are at top of the hour right now. It's about a minute until 12. So unfortunately, we won't have time for any additional questions. But if you'd like to learn more about this program, please don't hesitate to reach out to us directly. Our contact information is available on our website as listed on the slide that you see in front of you. And also make sure to check your inbox for the webinar recording within the next week. and you'll also have an opportunity to reach out directly to us as well. So thank you again, Dr. Popat and Dr. Sainani, for joining us today, and we wish all of you the best as you pursue your education goals and academic endeavors.
Thank you for joining us.
Thank you for joining us.